Hi, everybody. Everybody here at Cabrini Marine Aquarium and everybody at home, hello. Welcome to Cabrini Marine Aquarium's Discovery Lecture Series. I'm Dr. Julianne Passarelli, the Education and Collections Curator here at Cabrini Marine Aquarium. I'm going to just start with some information for the online viewers. So those of you that are here in person, bear with me for a moment. We are also streaming this live as a webinar for a bunch of people that are at home. So I want to give you guys at home some information that this is a webinar style platform. So you can't see yourself, nor can you mute or unmute yourself. So don't worry about accidentally making sounds during the talk. But if you have questions, please put them in the Q&A feature. Typically, it's on the bottom of your screen. We'll get to the questions at the end of the lecture, but during the lecture, if you think of a question, go ahead and type it in there. Put it in the Q&A, like I said. We're also going to be monitoring the chat, which is a different feature. Please use the chat for any specific questions about the aquarium, but the questions for the speaker go in the Q&A because then it's really easy for us to check them off when we go through the questions. Okay. Cabrini Marine Aquarium is owned and operated by the City of Los Angeles Department of Recreation and Parks, and we're extremely grateful for the city's support. I'd also like to thank the aquarium director, Chrislyn McCarran, and the program's director, Jim DePompe, for their support, and a huge thank you to everybody who has joined us tonight. The aquarium is open Tuesday through Sunday from noon to 5. All capacity restrictions have been removed, and we have resumed normal programming, and we're following all City of Los Angeles Health Department requirements. We have some upcoming programs that I would like to share with you. It is tide pooling season, so we have some public tide pool walks coming up. There's one tomorrow, Saturday, October 8th at 1:45. And there's also another one the next day on Sunday, October 9th at 2:15. Then there's a bunch in November and December. They're all listed on our website if you'd like to check that out. There's no need to sign up for these tide pool walks. Just come on down and join us at the aquarium, and we'll walk out to the tide pools together. We also have a public fish dissection coming up on Sunday, October 16th at 1:15, and another one at 2:15. We do 2 sessions. And this is where you can get a closer look at the outside and the inside of a fish. We also have a Walk Cabrillo coming up on Sunday, October 23rd at 1 p.m. You can pack a snack and join a walk of the Cabrillo Coastal Park guided by CMA's education staff. I am pleased to announce that the last of our 2022 Discovery Lectures will continue to be in person at the aquarium and will continue to be run as a hybrid lecture. We're going to keep doing that until nobody shows up at home and everyone shows up here, but we will continue to do it hybrid. Our next lecture will be on Friday, December 2nd at 7 p.m. Our speaker is Dr. Douglas Fudge from Chapman University, and the title of his talk is How to Survive a Shark Attack, Lessons from Hagfishes. So please join us for that. At this time, I'd also like to acknowledge one of our educators, Alfonso Montiel, who is running the webinar. This is a very stressful job, and he does it so well. So round of applause for Alfonso. Okay, and then I'd also like to acknowledge our videographer, our photographer, Zach Rich in the back. He's recording all of our lectures, and we archive all of them, and they're all on our website. So thank you, Zach Rich. Okay. Okay, so so just so everybody knows that the lectures are always posted on our website. You perhaps get emails from me, but as soon as we know the dates and the speakers, we get them on our website, and then our Eventbrite site goes up, and you can RSVP either for the webinar Zoom link or to RSVP in person, and then you can get the parking pass. After our December lecture with Dr. Fudge, we'll have lectures in February and in April with Dr. Andres Carrillo from Antelope Valley College and Beth Schaefer from the Los Angeles Zoo. And like I mentioned, if you've missed any of our lectures, not to worry. They are all recorded and archived on our website. On the Discovery Lecture Series page, just scroll down to the archives, or you can go to our CMA YouTube page and they're all there. Again, please RSVP so you can get the Zoom link if you're going to watch it from home, or you can get the parking pass for the night's lecture. All right. Does it look okay, Alfonso? They can see that at home? Great. Okay. It is my pleasure and honor to introduce tonight's speaker, Dr. Jennifer Burniford from California State University, Fullerton. Dr. Burniford is a professor of biology at Fullerton. She earned her bachelor's degree from Dartmouth College and her PhD in marine ecology from Oregon State University. She is a principal investigator in the multi-agency Rocky Intertidal Network, also called MARINE, which is a collaborative group which conducts long-term monitoring at more than 80 sites along the North American Pacific coast. She has explored and studied rocky shores around the world for more than 25 years, from northern Washington state to the Baja Peninsula to New Zealand and Chile. The title of tonight's talk is Rocky Intertidal Engineers. Who are they? How do they do it? And why does it matter? Thank you, and please help me welcome Dr. Jennifer Burniford.
Thank you. Hello, everyone at home, and hello, everyone here. Can you hear me with the, is the mic sufficiently loud? Great. All right. Is that going to work? So every scientific project takes a village. And so I want to start out by thanking people so that you know the whole time who's been helping. I'm going to talk about several projects during this talk. The kelp work was conducted with collaborators from Cerritos College and a former undergraduate student at Cal State Fullerton, V. Nguyen, funded by the National Science Foundation, Cal State Fullerton, and the University of Washington Friday Harbor Labs. The owl limpet work I'm going to talk about was part of a master's thesis for one of my students at Cal State Fullerton, conducted in collaboration with Luke Miller at San Diego State. And then we got a lot of assistance from, well, Squeakers will feature prominently, the Living Coast Discovery Center, the LA County Natural History Museum, and a collaborators lab at Cal State Fullerton, Misty Paik-Tran. The mussel work that I'm going to talk about is funded by Bureau of Ocean Energy Management, and our local site here is also funded in part by Cabrillo Marine Aquarium and the Friends of Cabrillo Marine Aquarium. We're very thankful for their help. Marine is a collaboration of scientists up and down the North American coast, and we are all independent workers that come together and use a standard set of protocols to do long-term monitoring. Some of our sites have been monitored since the mid-90s. The same protocols, different teams up and down the coast, so we get a very large-scale look at rocky intertidal systems. And my local work is done in collaboration with Jason Smith at Cal Poly Pomona. So today we're going to talk about ecosystem engineers, which was a group of organisms important in all systems that was defined by Jones and colleagues in the 90s. So the classic examples are trees and beavers. We'll get to the marine stuff in a few minutes. So ecosystem engineers modify, maintain, and create habitats. And they do this by altering the quality, the amount of resources, or the way that the resources are distributed in the environment in ways that affect other species. Just being eaten or eating something else is not considered engineering. So, and ecosystem engineers have a big community impact. They might individually have a big impact, but even if they have a small impact individually, they together as a population have a large impact. So for example, the environment under a canopy of trees is shady, it's moist, it's less windy than the outside. The trees, the structure of the trees creates an environment that's different in that local area as opposed to a few meters away outside the tree canopy. Beavers are ecosystem engineers by their behavior, and so they cut down trees and they move the trees into a different area. They change the flow of water in a way that affects the availability of water and the localization of water for other species. So if you're familiar with marine systems, you've probably heard a lot about the term keystone species, and so I just want to make it clear from the start how these are different. Keystone species are those that have individually a big impact. So yes, they do have a big impact as a population, but that's in part because each individual has a big impact. And for being a keystone species, being eaten or being the eater is one way that you can be a keystone species. So the very first keystone species, and the classic example, is Oystercatcher, the ochre sea star. And they play a keystone role because they eat mussels. And each individual sea star can eat a very large number of mussels. And just by the act of eating mussels, they exert an influence on the community that then makes them a very important player. So a community an area that has sea stars looks very different than an area that doesn't. I've been helping my 7th grade niece with her math homework a lot lately, and so I've got Venn diagrams on the brain. So ecosystem engineers are a group of organisms, including trees and beavers. And you can be, well, maybe not you, an organism could be both, a species could be both. So beavers are both ecosystem engineers and keystone species because they exert individually very big impacts on the community, and that the impact that they have is through engineering. Oystercatcher, though, the influence that they exert is only through eating other organisms, so they don't count as engineers, but they are, since they're individually important, they are keystones. The species that I'm going to talk about today, mussels, kelps, and owl limpets, these are all ecosystem engineers, not keystones, but still very cool and very important. So the rocky intertidal zone is where I spend a really large amount of time, is an extremely intense environment, just as intense as any desert, and way more intense than I think a lot of us think about on a regular basis, because if you go out into the rocky intertidal zone, you're usually out there, well, if you might be in that area diving or snorkeling, but you're usually out there when it's terrestrial conditions, which is what's great for us. So the rocky intertidal zone is the area where the land meets the sea. So the land, constant terrestrial conditions, exposed to air. The subtidal zone is the area that's always underwater, constant marine conditions. And the area in the middle is the intertidal zone, and that's the area that's affected by tides. So at low tide, it experiences terrestrial conditions. And at high tide, it's under full strength seawater. Now, the organisms that live in the intertidal zone, they all evolved in the ocean. So when it's full strength seawater, that's where their ancestors all were in those environments. But during the low tide periods when they're exposed to the terrestrial conditions, that's a very stressful time for them. And that's when they have to be able to tolerate these kind of changes, which are extreme in and of themselves, and also when you think about the ability to live both in full strength seawater and in air with the sun and the accompanying radiation exposure, which can cause, ultraviolet radiation and intense sunlight can cause damage to cells. And then you've got the rays of temperature as well from absorbing that radiation. And then you've got wind, and they're drying out. These are my very fancy marine organisms. That can be very stressful for them. So this is the time that I'm really interested in, how they are able to survive during these potentially very stressful periods. So for example, to get an idea of the stress, Dana Point, low tide, January 2015. Water temperature is 17 degrees Celsius. Limpets on the shore on that day were experiencing temperatures up between 36 and 40 degrees Celsius. All right, so our bodies, of course, a very small change in our core body temperature, and that's really bad for us. But these are ectotherms. These are organisms that their body temperature is entirely dependent on the environment, and they are going from 17 degrees up to, say, 40 degrees, and then being plunged back in 17-degree water when the tide comes back in. So it's very, very stressful for them. And this can be exhibited, well, it can be really seen pretty easily on a warm day. So this is a low tide. This is in Washington in the summer. And you can see, hopefully, the steam rising from the kelp here. So the water is evaporating off, and the kelp is becoming crispy. Here are 2 kelp individuals. We were putting seawater on this one every half hour during the tide. I love algae. They don't always photograph very well, but this is a fabulously healthy brown kelp. This guy over here is not being watered. It's drying out. It's becoming crispy, like literally the tissue is becoming crusty, dried out. And then when the water came back in, what you can see is that as the water comes in, you've got these green tissue patches because the tissue is dying, and then that tissue will slough off and float away. So it can be very, very stressful for them. Okay, so the first ecosystem engineer I'm going to talk about is this kelp, Hedophyllum sessile. It's commonly known as the sea cabbage, and so that's because each individual looks quite a bit like a cabbage. And I study this up in Washington State. I started doing this work for my PhD years ago, and I still work on this species. Even to now, I still go up in the summertime, and I work at Friday Harbor Labs in the summer. So we've got the kelp. Each individual kelp has multiple blades, and as the sun hits the kelp, it blocks sunlight from reaching the rock surface. So on a typical day in July, if the water is 10 degrees Celsius and it's an afternoon low tide, I've recorded temperatures up to 32 degrees Celsius in the open, but only 18 degrees Celsius in the shade. So in case you were wondering, it's cooler in the shade than it is in the sun. But this is a really substantial difference in temperature over just a couple of, a few centimeters. And so this can be very impactful for the organisms that live on the rock surface with the kelp. Also, a single blade of kelp blocks more than 98% of the light wavelengths. So these are wavelengths across the board from 300 to 700 nanometers. So that includes ultraviolet radiation A, UVA wavelengths, which are damaging. And then also the wavelengths that are used by other seaweeds for photosynthesis. So it's dark across the board. It blocks light across these wavelengths. And it also blocks some of the UVB radiation as well. So that radiation reflects off the kelp or is absorbed by the kelp, whereas that radiation is very intense just a few centimeters away. And so this is the engineering impact of the kelp. It creates these localized environments that affect other species. So over time, I established plots that were about the size of an 8.5 by 11 piece of paper. And I had 25 plots, and I counted out, after a year, I counted up the number of species of seaweeds that were in plots outside the algal canopy, or outside the kelp canopy, and generally there were, on average, 14 species of algae out there. And underneath the kelp canopy, there were only 7 algal species. It's darker. The species that live under the kelp canopy have to be able to survive in very low light conditions, but the organisms outside the canopy have to be able to survive in very high light conditions. So it's really a different set of adaptations that are necessary to live in these two areas. My very detailed algae. And then, so what about the animals? Well, I routinely searched areas outside the canopy and inside the canopy. So I searched over many, many days, over a few years. And out of the 400 sea stars, this is Leptosterius, the northern six-armed sea star. And for every one individual I found in the open, I found 81 individuals under the canopy. So many more of these are found underneath the canopy as opposed to in the open. Pisasterocratius, the keystone species. Again, they are far more abundant under the kelp canopy than in the open. So of the 935, most of them were found under the kelp canopy. Sea stars actually have sensory cells at the tips of their arms, and they can detect light. And so they will, in fact, I did an experiment with Pisaster, And when the tide comes in, when the water is low, they will move around and find shade and move into shady areas as the tide is going out. And this is a way that they are then protected from damage from radiation and from increasing heat when the tide is low. Chitons, who are herbivores, and snails are also similarly much more abundant under the kelp canopy. Again, and yes, these are organisms that can move, but I've tracked individual organisms. So for example, I put tags on these individuals, and there are some chitons that in an area where there was a lot of kelp, I found the same chiton in the same 1 meter by 1 meter square area for 4 years running. Every time I'd come back, number 64 was just right there in that same little plot. So they could move away, but the evidence suggests that they don't always. In many cases, if they're under an area of kelp canopy, they'll stay there for years. So the area underneath the kelp canopy is composed of a highly diverse set of organisms. I mean, I talked about some of them, but we've also got little snails, other species of sea stars, isopods, a lot of chitons, all concentrated under this kelp canopy. And that's really different from the environment outside the canopy. Some species are found in both areas, but at different abundances. Okay. Now, this isn't good or bad. It's not that one area is better than the other. It's just for some species, the habitat under the kelp is, they benefit from the cooler, shadier environment. But for other species, it's a negative effect because they don't have enough light to photosynthesize. So it's the structure of the kelp that changes the conditions and has a very big effect on the community. So ecosystem engineers, one single individual of kelp isn't going to do much, right, for a big rocky intertidal area. But multiple individuals of kelp together can provide a canopy that is fairly consistent over time. So we looked at the size of individuals as a way of looking at how much that canopy might change over short periods. So we measured the blade lengths. And at the beginning in July, we measured 6 individuals that we had tagged. And what happens is, as I showed you before, these individuals get kind of crispy. The tissue starts to get degraded, and then the tissue will come off as the tide comes in at the end of the day. That tissue will slough off, and the individual will be shorter. So we showed over time. On July 2nd, on average, they were 11.3 centimeters blade length. In the middle of July, they were 8.42 centimeters. And so as this happened, repeated hot, dry days meant that every individual kelp was getting smaller and smaller. Okay? And so, but if they're in a big patch, they can still provide a canopy. That one solitary kelp that's out there is going to continue to lose mass and not be a very big impact on the community. I did this in part because over repeated years working at this field site, I noticed that we started to see a really big difference. Between some years, we had really high cover of kelp, so all of this brown, fairly pixelated area down here, this is all covered by hedophyllum. Other years, this is exactly the same view. Here's this rock up here. You see all this bare rock down here. And in fact, none of this is kelp. This is all a rockweed that has moved down into the intertidal. So in other years, the abundance of kelp is extraordinarily low. It turns out that this is due to a natural cycle that is driven by the angle of the moon with respect to the equator of the Earth. And that determines how many hours the kelp is exposed to air. On one extreme at this site, individuals experience more than 200 hours exposed to air. And at the other end, it'll be much, much, much lower than that. And so these natural cycles mean that you have big changes in kelp cover, which then has really big changes or really big effects on the rest of the system. And that's, again, through the ecosystem engineering of that kelp. Okay, so the next individual, or the next species, is Middleus californianus, the California mussel. And mussels are mollusks. They have a fleshy body, and then they have two shells that they can open and close. At high tide, they suspension feed, so they pull food from the water column. At low tide, they can close up the shells to prevent them from drying out. That's a trade-off, though, because closing up the shells means they're not likely to dry out, but then they also can't use evaporative cooling, and so they can get very warm. Mussels can create fairly dense beds. This line here is because a wave has come up and washed the individuals that are darker here. They can create fairly dense beds because they have byssal threads. They have protein anchors, which they can use to attach to each other, and then also to the rock surface. So here are some byssal threads. There's some in this corner. Middleus californianus, which is very common down in this area as well. It's common all up and down the North American Pacific coast. They are pretty, well, they're cool in many ways, but not only do they create these byssal threads at high tide, but they can also create byssal threads at low tide. So here's an individual, my graduate student, Georgette, was doing some work looking at the effect of heat stress on byssal thread production. So here's an individual that has some byssal threads already, and this is the individual's foot that is extended, and you can see the foot kind of moving up and down a little bit, and that's because what happens is it makes its foot into a U, and it pours a polymer down, and then that polymer hardens, and it attaches with a plaque at one end, and then now it has a byssal thread that is basically an anchor, a protein anchor, to whatever surface it's on. So again, mussels are ecosystem engineers, and they can attach to each other and the substratum with their byssal threads. And so how do they modify the environment? Well, at high tide, if you look at the velocity of the water flow that up and outside of the mussel bed areas, and you compare that to the water flow inside the mussel bed area, the water flow inside the mussel bed is far lower. It can be 0 to 10% of the velocity of the water outside of the mussel bed. And this is important because when you have a lot of juveniles in the water column, a lot of marine species, when they reproduce, their juveniles float. And in order to settle and grow up into an adult, they have to be in an area where the water flow is slow enough that they can drift down to the surface and then have time to attach before the water pushes them away. And so the fact that the water slows down in mussel beds is really important in order for them to be able to settle, in order for organisms to be able to settle out of the water column. At low tide, the mussel beds again create an area that's very different in terms of the conditions than the area outside. So inside or outside the mussel bed, this study was done in Bodega, 37% relative humidity, warmer than 35 degrees Celsius, less than 25 degrees Celsius. So again, it's cooler in the shade, and when it's shady, it tends to be a little bit more humid, but this can be really important because over a very small scale, the mussels are creating an environment that's very different that will allow organisms to survive, or maybe they can survive on the outside, but inside the mussel bed, it's cool enough that they have enough energy, they can grow more, they can reproduce more. It's a very different environment. So inside the mussel bed then, you've got a variety of organisms. As you can clearly see, these are barnacles, my fabulously detailed barnacles. So you can have organisms that'll be able to settle there, again, survive, grow, reproduce. Inside the mussel beds, then you can also get sediment that is trapped, and so the trapping of sediment then creates a little sedimenty area that's different than the rock surface. And then you can get things like worms, again, my very detailed worm, that can live in the sediment. And so as the mussel bed exhibit in the aquarium shows, if you haven't seen it, you should check it out. There are, you can get a lot of species that are found inside these mussel beds. So in 1978, Cantor did a study looking at 20 sites up and down the Southern California Bight. Each site, he took cores, 5 cores, 300 centimeters in area. And overall, he counted 610 invertebrate species in these cores from mussel beds and 141 algal species. So this doesn't mean that these are species not found on the rocks, but what it shows is that in these conditions you have, it's not just mussels in these beds, it's a lot of other species as well. So in this photo, we've got several species of barnacles on, so these are acorn barnacles on the mussel. We've got Polycipes polymeris, the gooseneck barnacle living among the mussels. There's a little chitin hiding down here. There's another snail, or the a snail up here. And so inside the mussel beds, there are a lot of species that are taking advantage of these conditions. In addition, there are a lot of species that we know can't grow under those conditions where the mussels grow. So this is a picture taken up north. This is the sea palm, Pastelsia palmiformis. And there's a really famous study from the 90s that showed that adult Pastelsia release their propagules, and those propagules settle and they go underneath the mussel beds. Now the mussel beds block light, and so those propagules can't, kelps have a fairly complicated life cycle. Those propagules stay in a dormant state because they don't have enough light to produce gametes and reproduce to create the next stage. But when the mussels are removed, the light that is then shown onto the rock surface triggers reproduction, the kelps grow, and you can get Pastelsia on the rocks. And so, again, just like with the kelp, the shade and the temperatures under the mussel bed are really good for some species, but it also prevents other species from growing there. Okay. So mussels, very important to the whole community, big part of structuring so that if you go out in an area that has mussels, the whole community is very different from an area that does not have mussels. Mussels, being is important and common, are one of the species that Marine, the multi-agency rocky intertidal network, it's one of the species that we monitor up and down the coast. So we recently wrote a paper that used data from more than 70 sites. And at each of these sites, we use exactly the same methods. And we go out once a year, and we survey fixed plots to monitor the abundance of species. Anybody that is interested in learning more about what we do, I'm really happy to talk to you about this group. It's amazing, and I'm thrilled to be a part of it. So we meet twice a year to make sure that everybody involved collecting these data is on the same page. We use the same protocols. We talk about how to make sure we're using them the same way. And being in this consortium means that we have data that we know was collected with the same standards, using the same methods. From up north in Olympic National Park, where the National Park Service team collects, monitors their plots, down through Cabrillo National Monument, where a different Park Service team monitors their plots, and everywhere in between. So when we looked at this study, we're going to talk about data where we divided up the northern range, so that's anything north of Bodega, the central area, which is Bodega down through Point Conception, the south area, which is Point Conception down to San Diego, and then the islands, Channel Islands, which we analyze separately because the oceanographic conditions are really different there. And in each of these areas, we had fixed plots, which have bolts at the sides. And every year, we go out there and we have a grid, and we record what's under every point in the grid. And we also take archival photos. And if you're here tomorrow on the tide pool walk, you'll see us, because we'll be out at the Point Fermin site collecting our data. Okay, so we looked at the cover of mussels from 2005 to 2017 for this analysis. And we used that range because that gave us the largest number of sites that had continuous data. Some of our sites go back to the 90s, but we had a large number of sites that were started in the mid-2000s. And so this is the average cover of mussels across all of the sites in this region. And you can see from 2005 up through 2017, in the north, so that's above bodega, and in the central area, the cover is relatively constant over time. But when we started to look at the sites down here, so the Channel Islands and more relevant to us, the southern sites, we noticed that in fact mussel cover was not staying constant through time, was declining fairly substantially through 2015, and then there was a little dip up. But the pattern that we saw in the south was definitely not the same pattern we were seeing in the north or in the central regions. So since I've been talking about how important mussels are for the, for the whole community, we also did an analysis where we looked at community similarity. And so what we did for this is we took data from 2006. We looked at every species that we encountered in our plots. And we not only looked at the, what species it was, but we noticed how many points we hit, so how common it was. And we did this for 2006, and then we did it for every site every year after it, so we could say how similar is this site to what it looked like in 2006. Okay, so if a site was really similar over time, if there was no change since 2006, we would see that the data from each year would be a straight line and 100%, which, spoiler alert, obviously isn't the case because our graph doesn't even go to 100%, but that's what it would look like on this figure. Meanwhile, if you have a site that was moving further and further away from the community assembly that was there in 2006, you would see the points declining over time. Because in this case, it's saying that in each successive year, the community is less and less like the community was in 2006. Okay. So overall, all of the, and these are averaged across regions. So in all of the regions, the north, the central, the communities were becoming more and more different from what they looked like in 2006. But the drop in the southern regions was much greater than the drop in those northern regions. So everything's moving away from the structure in 2006, but in the southern sites, the Channel Islands and the mainland, the pattern is really exaggerated. And remember, these are the areas where the mussels were declining quite substantially over time. So we spent a lot of time trying to figure out what might be responsible for these changes over time. One thing that we did notice was that years where you saw the biggest change in the south were associated with years where the minimum water temperature wasn't getting as cold as it used to. So, it's easy to say, oh, it's a temperature effect, but then you have to find out what the temperature effect is. And the data that we analyzed indicates that the biggest changes are in the regions and the years when the winter water temperatures aren't getting as cold as they used to. So this is something that we're going to keep trying to figure out why this is happening. But it's really important to be aware of the trends and to know that this is what's happening in natural systems, especially because, as we all know, unexpected events occur, and sometimes humans can come in and make changes that are layered onto the top of these natural changes. So for those of you that are local, you may know White Point, just around the bend in San Pedro. So these are data from the plots that are surveyed. They used to be surveyed by UCLA. Now they're surveyed by Cal State Fullerton and Cal Poly Pomona. We saw the same pattern of a big decline in mussel abundance between, sort of around the 12, 13, 14. Looked like it might be starting to come back at this site, and then you'll notice this big dip between 2019 and 2020. That was associated with the onset of COVID, and particularly at White Point, we saw a really big increase in the number of people coming to the intertidal zone and the number of people making collections. And this is, we don't know if they made collections in our plots, but this pattern that occurred right here in fall 2019 is associated with a substantial drop in mussel cover around that time. So as we know, ecosystem engineers they exert their influence when they are abundant in the, right, as a population. So it may be that a drop of 20% mussel cover maybe wouldn't really be a big factor in the community if mussel cover was really high. But when mussel cover is already very low, this abrupt shift may result in big changes in the population, or not, I mean, obviously in the population, but big changes in the community moving forward. So it's just really important to understand the ecological role of different species so you can figure out, is a trend like this going to have larger impacts on the community? Okay. Our last ecosystem engineer is the owl limpet Ladia gigantea, which exerts its influence as an engineer through its behavior. So now we're doing a little bit more moving than the kelp and the mussels. And so it's more like the beaver because it's what it does, not just its structure, that has big influences on community structure. So owl limpets are territorial mollusks. So here's this owl limpet, and this is its territory. And they use their feeding apparatus and their shell to bulldoze and remove algae and invertebrates from these areas on the rock surface, making them, as we know, hopefully from 5 minutes ago, an area that's warmer and has a lot more light and is less humid than the area nearby under the mussel beds. And so owl limpets can actually physically fight with other limpets to get them out of their territories. They really do defend their territory with very intense battles. I spent a lot of time in the intertidal. And then they graze the very small microalgae, small, that grows in these open areas. So here are the radular trail marks of this limpet that excludes other species from the area and then has this little garden of algae that it feeds upon. So again, one single owl limpet at a site, not really going to make a difference. Lots of owl limpets at a site, you're going to see, so in this case there are 3 territories, that's going to start to have an effect on the larger community as you look across the site. So these 3 limpets have individual territories. Now, don't feel too sorry for the mussels. First of all, the mussels are very important for juvenile owl limpets. Here you can see we've marked them with lumber crayon. Lumber crayon washes off when the tide comes in. This is how, when we monitor their abundance, we make sure we don't double count them. We mark them as we count them, and then it totally washes off as soon as the tide comes in. So juvenile owl limpets live on mussels. They have little territories on individual mussels sometimes. And then actually, one of the causes of adult owl limpet mortality is if they nestle in too close to a mussel bed. As we saw, the Middleus californianus can get their abyssal threads out with relative speed. It can be, I think they can make them in as little as 15 minutes. They can create a abyssal thread. And sometimes they put abyssal threads on the limpets, and then the limpets get stuck. And so then they can't move, they can't feed, ultimately they die. Now, mussels aren't doing this on purpose. I love the intertidal organisms, but I don't think they're out to get each other. They don't have intent. But it's not like owl limpets are going to denude the intertidal of mussels. Okay. And other studies have shown that if you remove owl limpets, the algal cover goes crazy. So they really do exert a very big influence with using their shells and creating this area that prevents mussels from moving in and prevents big algae from growing up there. So way long ago, I showed you this slide of a typical day at Dana Point where you had very hot limpets that were out there on the rocks, and the water temperature was much cooler. Well, these were actual temperatures that we collected using robolimpets. This was my student, Rachel, as part of her thesis. So robolimpets are a type of biomimic data logger that we actually use by taking a shell, a found shell. We never killed an owl limpet for this. You take a shell, you put a sensor inside, a temperature sensor, and a chip, computer chip that'll record the data. And it's been shown by other people that this actually gives you accurate body temperatures for the limpet. And then you literally stick them on the rock surface, and they take data for you 24/7. And you know that you're getting data that are accurate and relevant to body temperatures of living limpets, but you don't have to poke and prod those limpets all the time. So we use these robo-limpets out on the shore, and that's how we know how warm these limpets were getting during low tide. So my incoming student, Rachel, said, oh, this is so interesting, the limpets get so hot. We know that heat can cause really high heart rates in limpets, and that might be a problem for them. We know that heat also can affect proteins and might affect muscle contractions. So, hey, why don't we look for, for her thesis work, she wanted to know, does heating up at low tide affect the susceptibility of owl limpets to predation by sea stars? So when sea stars approach an owl limpet when it's back in the water, the owl limpet will run away. It has a very elaborate escape response. So we were wondering, does being heated up at low tide, does that impact the escape response? And maybe the limpets are not able to escape from the predator when it comes hunting for them when the tide comes back in. So this was great. We had the limpet data. We were all excited to do this. And then all the sea stars disappeared from sea star wasting disease. So we thought about it for a while, and we said, okay, why don't we study how low tides affect susceptibility to a different predator, the black oystercatcher. Neither of us were bird people, but we had to roll with it. And so we decided to go down an entirely new path, looking at how black oystercatchers might affect or might be able to eat limpets, and whether being exposed to warm temperatures at low tide might change this interaction. Great, okay, so when oystercatchers attack a limpet, they hammer it with their beak, they flip the limpet off of the rock, and then they eat the soft tissue from underneath the shell. We knew that. We thought, great. So the first thing we need to do is figure out how to make a mimic of that beak so that we can see how easy it is for an oystercatcher to remove the limpet from the rock. So what we did is we went to the Natural History Museum, and we looked at specimens of oystercatchers in their collection and measured the beak parameters so that we could build a very, very highly, a mimic that exactly mimicked the measurements. So we made a mimic, we printed it on a 3D printer. Well, my colleague, Missy Peg-Tran, and her grad student printed it on their 3D printer. And so then we had a beak mimic that was literally the shape of an oystercatcher beak. And then we made this bird mimic. Looks exactly like a bird, right? All we cared about was the beak. And we put the beak mimic on a spring scale, and it has this hook, so it's basically a pool cue effect. We know that when you pull back on this hook, it compresses a spring to a known amount, and so we would set it here, we'd pull it to exactly the same amount, and when you let that go, it springs out with a known amount of force. And so we knew that this is how we wanted to mimic the bird attacking the limpet. So we thought, how do we mimic predation in the lab? We were all over this. We do this kind of stuff all the time, no problem. We were going to put our owl limpets on tiles, and that way we could let the owl limpets stick on the tile, and we could move it around into different treatments and still allow it to attach, so that then when we hit it with the pool cue, we knew that it wasn't being affected by us moving the limpet around, right? It could attach to the rock just like it could in real life. So we had a hook, our spring scale on the bench. We were going to put our limpet on the tile. We had a camera. We had all this stuff. We were all great. I mean, we didn't set this up, but we had the whole plan. And then we thought, great, okay, well, what angle do we attack the limpet at? What side do we attack? Do we attack the limpet from the front? Do we attack it from the side? Do we attack it from the back? where do oystercatchers attack limpets? We started asking all these people that had worked on oystercatchers and limpets before, and everybody said, I don't know. So we had this whole list of questions, and then also, of course, how much force do they use? How should we mimic predation in a realistic way? All right, because it's not about how much force we need to, we need to push an owl limpet off the rock. It's how much force can an oystercatcher use to get the limpet off the rock. So my student said, I have a friend who works at the Living Coast Discovery Center, and I think they have an oystercatcher there, the bird. That's what they called it, the bird. So Rachel talked to her friend at the Living Coast Discovery Center, and it turns out they did have an oystercatcher. And Rachel said, do you think we could maybe feed the oystercatcher some limpets to see, to get information about how it attacks them and the angle and the force and everything? And her friend said, yeah, that's great, except this oystercatcher was raised in captivity and has never eaten a limpet before. So Rachel's like, that's not a problem. She goes, she puts a limpet out on a tile to the oystercatcher, which just completely ignores it, because I don't know if you've seen, it's like, why is this lumpy tile out here? Like, it didn't even register it as food. So Rachel spent several months teaching the oystercatcher that limpets are food. So first she started with upside down limpets so that the oystercatcher knew, hey, this is cool, and then she'd turn the limpets over, and then it'd be like, oh, hey, if I can turn this over, there's some food under there. And she spent several months training the oystercatcher to recognize the limpet. So it's essentially like a juvenile oystercatcher. Oystercatchers learn how to hunt. They learn from their parents. They learn what are food, what's not food, and then how to attack them. So it was essentially going through this whole thing with Rachel as the parent. And so the bird got pretty good at removing the limpets, and they were doing this over a few months. And then Rachel would go out and take high-speed video of the bird. And finally, the people at the center thought, okay, this is ridiculous. We keep calling it the bird, so they named it Squeakers. So Squeakers got a name, and then Squeakers continued after we trained it to use limpets as food. Then we were able to move forward with the project. So then, of course, we have all our questions. Where do they strike them? What's the angle of attack? How much force do they use? And we called up a colleague that I knew from some meetings and things. And we said, Luke, are you interested in working on this project with us? And he said, absolutely. And he just flat out worked with Felicia, and they're like, here's a force meter. So they built this force meter, which we attached a limpet shell to the top of this thing. And then we put it in with Squeakers, who was happy to attack this limpet. And the limpet is attached to a meter that can record the direction of hits and the force with which the oystercatcher attacked the limpet. And so then we could get all of our data that we were interested in using. Side note, I've tried to use this with real oystercatchers in the field. This box is about the size of a laptop, or a little bit smaller, the size of an iPad. And so I take it out to the field, put it in the field. Regular field oystercatchers walk right by it. They're like, whatever, that's too flat, I'm not doing that. So then I'd move it, and then they'd just walk right past it again. So we have not been successful at getting them to use it in the field, but at least we have one oystercatcher, which is more information than we had before. So we found that this is a diagram showing the direction of push. So most of the attacks that Squeakers made on the limpet were from the front end, pushing it towards the back occasionally, but it did attack from all over. So there we go. And so we decided to use an angle of 35 degrees for our lab experiments and to hit our limpet in the lab from the front to mimic the majority of the strikes that Squeakers made on the artificial limpet. We learned that Squeakers had two basic kinds of behavior with the limpets. For pecks, these are less than 2 milliseconds, and this would be a rapid, usually high force attack on the fake limpet, on the meter. And then occasionally he would use pushes, which were slower, still less than a second, but a little bit longer and a little bit less force. Maybe he got tired, we don't know. And then we also learned that he almost always attacked multiple times in rapid succession. So we decided for our lab experiments to use 14 newtons as our fixed force because we thought that was relevant to both pushes and pecks. And we decided to attack our limpets in the lab 10 times and see could they withstand 10 14 newton pecks and still stay on the tile. So we had 3 treatments, no low tide, we kept them in seawater, a cool low tide, 14 degrees Celsius, and a warm low tide at 30 degrees Celsius, which is, completely relevant for what we see in the field, not even the hottest of the hot. And up to 10 strikes with our bird mimic. From the treatments we had, from the no low tide treatments, only 1 of 10 limpets was removed from the tile. The other 9 were still on the tile, still attached strongly after 10 pecks. The cool low tide, 2 of them were removed. Again, the other 8 still holding on strong. Individuals that had experienced the warm low tide, 8 of them popped off with these attacks. So it looks like there is really a big impact of low tide temperature on the ability of the limpets to hold onto the rock surface and thus resist getting eaten. We did the same thing with Squeakers. We presented him with limpets that with almost the same temperatures, but it's hard to get exactly the same temperatures because this was at the Discovery Center. So in this case, our cool low tide was 16 and our warm low tide was 32 degrees C. We had 30-minute trials, which is a really long period of time for an oystercatcher to interact with a single owl limpet. So Squeakers actually ultimately removed all of them from the tiles, because after 30 minutes he'd sometimes walk away and then he'd walk back and then he'd keep pecking at it. But when we looked at it, how long he interacted with them, two of the 9 limpets in the no low tide treatment were removed with less than 3.5 seconds of interaction. For the cool low tide, 4 of 7 limpets, again, were removed within 3.5 seconds of interaction. And for the warm low tide, within 3.5 seconds of pecking at them, he had removed almost all of the limpets. So again, in a situation where there are lots of limpets out there, it's going to be faster for an oystercatcher to pop off and eat a limpet that has been exposed to a warm low tide versus an individual that's been exposed to a cool low tide. So for any individual limpet, you could say it might be a much greater chance of getting removed and eaten if they've been exposed to a warm period than if they've been exposed to a cool period. And this is relevant both for changes over time, but also for microhabitats on one day. So a limpet that's on the shady side of a rock might be less likely to be eaten by a bird than a limpet that happens to be on the sunny side of a rock. Okay, so do warm low tides affect susceptibility of owl limpets to predation by black oystercatchers? Our data say yes. And this is really interesting to us in general, hopefully to you. But because at the time that we were doing this study, even though we knew that there was a lot of potential habitat for black oystercatchers, we didn't really see a lot of black oystercatchers in Southern California at this time. So the orange spots are locations where there's good habitat for oystercatchers at the time of this study. The purple spots show where people were seeing black oystercatchers. So they were being observed and they were actually foraging at a very oystercatchers are really susceptible to human interference in their nesting. And so anecdotally, people were saying that they used to see a lot of oystercatchers around in the 80s. Their numbers really dipped in the 90s and 2000s. But now we're starting to see them back here. So I've recently seen, over the last 6 months, I've seen a pair of oystercatchers foraging out here at Point Furman multiple times. I never saw oystercatchers out here in the 10 years before that. And I'm seeing oystercatchers at a lot of sites in Orange and LA counties now where I hadn't seen them before. So oystercatcher populations seem to be rising, which is great. But that also means that we might see unexpected changes in the community if they are consuming owl limpets, and if temperatures, warming temperatures, may affect this interaction. So we might see different outcomes of the presence of oystercatchers relative to periods that were generally cooler when oystercatchers were around. Okay, so hopefully I've shown what intertidal engineering or what ecosystem engineering is, and for kelps, mussels, and owl limpets, how they do it. I've tried to talk about why it matters for the ecosystem, but of course, it also matters to us. Hopefully, that's why you're here. So why do I think it matters to people? Well, I think it's really important to understand how the systems work so we can identify things that are driving changes in ecosystems, and we can distinguish the driver from the effect, right? So we need to know that something that affects canopy-forming seaweeds is going to have a trickle-down effect and affect a lot of other species in the community. We need to understand which species are going to have big impacts and which species are going to have smaller impacts. And this is how we're going to be able to make predictions about changes, and we're going to be able to target our efforts so we can monitor species and we can track those changes over time. It's also really important to understand how systems work so that we're not vilifying native species, especially when things are changing, right? So oystercatchers are coming back in. They eat a lot of different types of limpets, but we might see that they're eating owl limpets and that's causing a change in communities. Again, oystercatchers aren't bad. Oystercatchers are a natural part of the system. And so it's really important to understand the different relationships between organisms so that we don't have sort of runaway, oh my gosh, this species is bad, right? It's not a judgment, it's just understanding the different ways that species interact. Knowledge is power. And of course, to promote informed and effective stewardship and, if necessary, restoration events, right? So knowing that ecosystem engineers have big power in large numbers, we might say, okay, if we're going to restore a kelp community, or we need to restore patches of kelp, not just one or two individuals, right? We need to protect patches of mussels if that's something that we need to do. And so again, we have to know how the ecosystems work in order to protect them, and if we need to restore them, we'll restore them in ways that are effective and efficient. Thank you very much, and I'll take any questions.
Thank you, Dr. Berniford. That was great. Can I just get a thumbs up from someone at home that you can hear me with this mic? Someone thumbs up? Great, okay. All right, so if you guys have any questions for Dr. Berniford, she'll be happy to take some. I'll let you call on.
Oh, sure.
I'm going to repeat the question, just so you guys know. When you ask her a question, I'm going to repeat it so the folks at home can hear the question.
Well, I was considering the 2013-14 drop at White Point, that big factor of 2. I would be interested to know what you think that was about, because that's clearly a very big event that must be some explanation.
Am I getting too close? I'm getting too close to the technology. So he's asking about the 2013-2014 drop.
Right, in mussels.
In mussels.
In fact, I showed you the data from White Point, but that's something that's across the South Coast region. That's a great question.
Nope.
Is that me?
It was something you did? We're good? We're good.
Okay.
Sorry.
That's an excellent question, and we are still trying to figure out what is associated with that. It was at multiple sites, and so it appears that it's some sort of larger, maybe climatic factor. And one might think, it's been several years. How come you haven't, why isn't that done yet? But you can only find a pattern if you look specifically for that pattern, right? So there are a bunch of people working on a manuscript trying to analyze these data, and they're looking at air temperature, water temperature, mussel body temperature, because we have robo-mussels out there. It's not just robo-limpets, we got robo-everything. So that's an excellent question that we still have not pinpointed the environmental factors that were associated with it, and it's an ongoing topic of research. And believe me, when we have found that out, we are definitely going to advertise it. So yeah.
Okay, so your first question. Your first question was about the removing owl limpets. How was that going to impact the community?
Right, because of the algal growth, I think.
Because of the algal growth.
100.
Yeah.
And then the second question was about the 2020 decrease
right.
because of COVID. Yes.
And you said it corresponded with the COVID time.
With the COVID time, yeah.
Right. Okay, so the owl limpet question. So we're talking about the potential for oyster catchers to decimate owl limpet populations. So I don't think that it's going to be possible that a single pair of oyster catchers, because I didn't say this, I forgot to say this, but oyster catchers are territorial as well. And so you usually get a pair that forages along a stretch of shoreline. It is unlikely that a pair would be able to remove all of the owl limpets from an area. And so, because there are lots of owl limpets, and oyster catchers eat all sorts of limpets. They eat all species of limpets, not just owl limpets. So it seems like it would have, I would predict, I don't know for sure, I would predict that it might have a patchy effect, and that we might see that difference in the warm sides of rocks versus the cool sides of rocks. We might get a more differentiation between those areas. The study that looked at the removal of owl limpets, it was a several-year pattern where algae would increase very rapidly and stay there for a while, and then we don't know what would happen after that. You might see mussels moving in, so it would be a rapid change and then a slow follow-up change. But I don't think that a pair of oyster catchers could remove all the owl limpets. It's a great question, though. The COVID, I was absolutely not trying to imply that the COVID itself was affecting the mussel populations. What happened was at White Point in particular, there were documentation of a huge number of people going into the intertidal zone and removing organisms in a way that people had not been using the shore previously. And so, I sort of think is happening, I've talked to colleagues up and down the coast, is, as we all remember, we were suddenly trapped in our houses, and so getting outside became a really big thing, and people were trying to figure out what they could do to get out and be active and be with family members and not be trapped in their homes. And in some areas, actually in many areas across the coast, this manifested as more people going into the intertidal zone than would be typical. We also noticed that there were lots of, and this is backed up by California Fish and Wildlife data, more people applied for collection permits to harvest for personal use. And in addition, there were many, particularly at White Point, there were many people that were cited for not legal collections. So it was a change in behavior. And I actually, I'm writing a manuscript on this. There were way more things we've noticed, a huge increase in, many of our things got vandalized. Our data loggers, our sensors in the intertidal zone got vandalized around that time. You'll notice that a lot of rocks are still getting tagged in the intertidal zone. People are spray painting on rocks in the intertidal zone, which never happened. I never saw before. So there seems to be, I think that was associated with just large numbers of people moving into the intertidal zone, not knowing the rules in many cases, just not knowing, or doing things legally and just increased pressure.
Jen, can you speak to what species had an increase in take either legally or illegally?
So I was particularly working with mussels, and that's a really good question. So through the Marine Network, I talk with CDF&W people very frequently, and they don't have data on, so I tried to get data for this manuscript I'm writing. They don't have data on the different, or they can't give out data on the different types of organisms, but we know for a fact that mussels were very strongly affected at White Point. There were 2 newspaper articles where people reported seeing, the Fish and Wildlife people cited people for carrying mussels. They saw people with hundreds of pounds of mussels. We saw people out there multiple times when we were collecting data, carrying mussels up and then dropping them with the Fish and Game Warden came in. And so mussels were particularly hard hit, but other species were as well.
I have a question. Are there any recent studies or interest in like the benthic engineers, or like at hydrothermal vents or anything like that, or studies like that?
So the question is about hydrothermal vents. Ooh, are there any studies on hydrothermal benthic communities?
Yeah, so subtidal communities and ecosystem engineers, absolutely. So kelp forests, right? The Macrocystis pyrifera, the giant kelp. I know that's not a hydrothermal vent. I'm going to get deeper. There are tons of studies that look at subtidal kelps and how they perform ecosystem engineering. And yeah, there are tons of people that are working on ecosystem engineers in all habitats, absolutely. It's a really interesting avenue of research that I really like, sort of like the general ecological topics applied to different systems. I mean, I don't work on hydrothermal vents, but there are ecosystem engineers in every system that I've ever read anything about. Yes.
Perhaps we'll do a few questions from home. Let's see. Oh, this one's good. Robo-limpets. What an incredible invention. How long have they been around?
That's a great question. So they were developed, to my knowledge, by the Wethy lab, Dave Wethy, on the East Coast, South Carolina. Although, if that's not who invented them, sorry, whoever invented them. They've been around for a long time. Definitely 2009, I know, is when I first discovered them. The thing is that in order to develop a robo-limpet or a robo-mussel, right, we have robo-mussels, it's not just slap a shell on it and call it good. You have to do lots and lots of testing under a wide range of conditions to make sure that your robo, whatever, really truly takes temperatures that match the body temperature of your organism in the intertidal. So yes, robo-limpets are great, and we take advantage of robo-mussels too. Through Marine, we've deployed robo-mussels at many of our sites up and down the coast to get these data. But it's great when a biophysicist figures out the model, and then we can deploy it as ecologists.
Okay, there's one more that I wanted to read here. This is from Octavio. Is there a website where students, teachers, or the public can find the data that Marine staff collects, and where can we find more information about the multi-agency Rocky Intertidal Network Group?
Excellent questions. Yes, we do have our data on our website. All our data are publicly available. That's one of the conditions with joining into Marine. We really want to make sure that our data are accessible. So on the screen here, I have our Marine Instagram, Marine_Pacific Rocky Intertidal, and that will give you a lot of information about the network. If you have Instagram, you can also get our website from there. But our website, which I should have put on here, is www.pacificrockyintertidal.org. So our Instagram is linked to our website. Yeah, and we've got tons of information about what we do, who we are, how you can get data. We have data sets available for teachers that they can use for education purposes. And and we have a citizen science page. We're very involved in sea star wasting disease research. And we have a citizen science page where you can put in observations if you see a sick sea star. That's how we were able to track, one way we were able to track the movement of the disease up and down the coast.
It's a great website. So there's some comments saying that they can't hear me when I'm repeating the questions. So
okay.
if you would, they can hear you.
Oh, okay.
So if you would repeat the questions, that would probably be better.
I can do that.
Okay, thanks, Jen. All right, more questions from the audience?
Great question. So the question is there any relationship between El Nino and La Nina and the patterns of mussel density and percent cover on the data that we analyzed for the 2021 paper? So yeah, the water temperature, the minimum water temperature in the winter, right, is associated in some ways with overall seawater temperatures with El Nino and La Nina, and that was factored into the analysis that we did. So I would suggest read the paper. It is open access, and I'm happy to talk to you about that later. And yeah, the water temperatures, those are things that we definitely looked at when we were trying to figure out the patterns. And I would definitely be happy to, so the paper is out there. Questions about the paper, definitely chat with me later. That's a great question. Okay, so those are really great questions. So the question is basically, so I'm assuming you're talking about the change in community similarity over time that we took with the marine data from the north, central, and southern mainland regions. And whether or not biodiversity was dropping, which was causing that change in community similarity. And then whether that's linked to foraging. So a site, so you could have, the way that we did this similarity analysis was it included both the identity of the species and the abundance of the species in this analysis. So if a species became less abundant in 2007, that would manifest as a change in similarity. It's not just if a species dropped out, it's if a species changed in abundance. The site would show up as having changed. We did show that that diversity did change over the same period of time. I'm trying to remember all the different factors in the paper. I used to laugh at my professors when I was like, how do you not remember this one thing in the paper? And now I'm that person that can't remember all of the separate analyses. So yes, diversity did change over the time, but we did not look at, I did not look at, and for this paper, none of us looked at the data set in a way to look at which particular species were pulled out of different areas, mostly because we had over 70 sites over such a big region. We didn't yet go in and say, is this species still present? Is that species still present? So that's a really interesting avenue for further study. In terms of whether foraging was driving the patterns, our sites are a variety of sites, including MPAs with very stringent access limitations and open areas that are not MPAs. And we found overall that the changes were occurring widely across the regions, that they weren't limited to MPAs or non-MPAs. That was a factor in the analysis, but it wasn't something that really drove the patterns. And when I talk about, I mean, some of our sites are literally, like, the public cannot get there. There is no foraging. So those are great questions, and we did look at some of them and not at others, and so that's a great idea for moving forward. That's a really great question. So the question was about whether the fact that sea stars are barely coming back in the south, they haven't really made it back, and so that might provide an opening for mussels to increase in abundance. And again, if oystercatchers are coming in and maybe altering the abundance of owl limpets, that might provide some room for mussels to bounce back. And that is a possibility. It kind of depends on what environmental factors are affecting the mussels. So I think a lot of us expected to see some bounce back in mussel populations down here when sea stars disappeared, although for that matter, sea stars down here were never particularly, I mean, at some of our sites, they weren't really abundant anyway, so we'd go out to, say, Dana Point, and we'd see a couple of sea stars. But some of our sites, we had a lot of sea stars, but now we don't see hardly any. And there may have been some little bit of bounce back, but that's layered on top of environmental factors that don't seem to be necessarily moving in a way that promotes a lot of mussel recruitment and growth. And so that's the thing, it's all interconnected, right? And so but that's the kind of thing we can make the hypothesis and then do studies to test it. So hopefully, we'll see some recovery in mussel populations because they are a very important species, but we can also collect the data on environmental conditions and the communities to try to see as it manifests. It's a really interesting question. So the question is comparing intertidal, so within a species, presumably comparing intertidal individuals to subtidal individuals to look at sort of why are individuals living in the intertidal zone when they could have a cushy life in the subtidal zone. So especially in rocky communities, it's two-dimensional, right? There's no chance to burrow into the sediment, and so there's a huge amount of competition for space. And even though the intertidal zone is stressful, it's also a place where if your predators are stressed out and can't really get into the intertidal zone as often, then it can be an area where if you can tolerate that, then you can get an escape from some predation. Sea stars, for example, don't move around and feed at low tide. They move around and feed at high tide. So it sort of reduces the chance for a predator attack if you're talking about low predators that move in the water. So the subtidal zone can be less stressful in terms of temperature and wind and desiccation, but it also presents more opportunity for predation by fish, by multiple species of sea stars, and things like that. Are they healthier in the subtidal zone? So they may be. In terms of if you look at individual health, you can think of physiological stress and then reproductive output, but also they don't have control, the larvae don't have wide-scale control over where they settle. They tend to settle out of the water column. They don't have intent. They can't be like, ooh, I know this is gonna dry out here later, so I'm gonna go down over this other area. The larvae can't choose where they settle, and so it's more about they happen to settle here, do they survive? But that's a really good question because it's, for mussels can't move around that much. So they settle, they live, or they die. So the intertidal zone can be a respite in some ways from biological factors that could impact populations in the subtidal zone.
I love that explanation, by the way. That's great. That's good. Any other questions? I have one. I want to hear more about this limpet battling.
So I don't actually do work on the limpet battling. I just have read a ton of papers and talked to people who do it. But limpets can actually be super active with their shells. I have done things like I threw a limpet in a tank of sea stars to feed the sea stars, and I noticed that the limpet, it was a tank, it was about 1 meter long, and I just looked over and the limpet was running around, twisting and turning, moving its shell, and there was a literal herd of sea stars following this limpet around the tank, and it managed to escape. I mean, they're wily and they have very good muscular control of their shells. So apparently the owl limpets will butt up shells with each other. They will move up and butt up into a limpet, and then it can actually push the limpet off the rock, and especially if this happens as the tide is coming in, the limpet will roll, lose its purchase, and move away. And so I haven't done studies, but I can definitely share the papers with you. I would love to see video too. Find a video, I'll show you. Yeah, they can move a little bit, but not, so I was comparing something like a chitin or a sea star, which can actually move directional for long periods of time, to a mussel. Mussels do have the capacity to move over small areas. They can unstick the inner byssal thread, leave the byssal threads there, move with its foot. So if sometimes you'll have a mussel in a tank, and you'll be like, how did that mussel get to the top of the tank? But it's very, very slow. It'll disengage, attach a new one, and they can actually sort of crowd in if a patch is opened up in the middle of a mussel bed. Mussels can kind of shuffle their way in, but they can't move, they're not gonna move meters in the intertidal zone. So yeah, they do have a limited capacity for movement. They're not sessile, they're sedentary. They tend to stay in the same area. Whereas something like a barnacle, it sticks and it doesn't move there at all.
Okay, well, let's end it with that. On behalf of the aquarium and Friends of Kabirma Aquarium, we would like to thank you so much for that presentation. So let's give Dr. Limpet a round of applause. Thank you. Thank you. That was amazing, Jen. That was amazing. Okay, so I'm just going to wrap it up with just a couple announcements and just a reminder. Thank you to Dr. Jennifer Burniford, and thanks again to Friends of Cabroom Aquarium and the City of Los Angeles for their support. Dr. Burniford mentioned that she'll be out sampling tomorrow at Point Fermin, and we'll have a tide pool walk, so you can join us for the tide pool walk if you like, and you can see her doing her research, which the Friends of Cabroom Aquarium has supported and funded. We apologize if we haven't gotten to all your questions, but we will be going into the gift shop, and Dr. Burniford will be there for a little bit of time if you have some more questions and would like to chat with her. Once again, the upcoming schedule and speakers for 2022 and 2023 is posted on our website, and the remaining Discovery lectures for this year will be in person, and we're going to run them as a hybrid lecture. And you can either come in or join us by Zoom on December 2nd. I'm getting tired, sorry. I'm losing my mind. And just a reminder, we've recorded this lecture. We are going to post it soon, and it will be on our YouTube page. So those of you at home, thank you again for joining us. Stay safe and hope to see you next time. And those of you that are here, please stay for our little giveaway. So thank you again. We'll talk to you soon.